The light-independent reactions of photosynthesis are chemical reactions that convert carbon dioxide and other compounds into glucose. These reactions occur in the stroma, the fluid-filled area of a chloroplast outside of the thylakoid membranes. These reactions take the products of light-dependent reactions and perform further chemical processes on them. There are three phases to the light-independent reaction, collectively called the Kelvin cycle: carbon fixation, reduction reactions, and ribulose 1,5-bisphosphate regeneration. Despite its name, this process occurs only when light is available. Plants do not carry out the Kelvin cycle during nighttime. They instead release sucrose into the phloem from the starch reserves. This process happens when light is available independent of the kind of photosynthesis. Can plants store malic acid in the vacuoles every night, and release it by day in order to make this process work? Coupling to other metabolic pathways, these reactions are closely coupled to the thylakoid electron transport chain, as reducing power provided by NADPH produced in the photosystem I is actively needed. The process of photorespiration, also known as C2 cycle, is also coupled to the dark reactions, as it results from an alternative reaction of the Rubisco enzyme, and its final byproduct is also another glyceraldehyde 3P. The Kelvin cycle, Kelvin-Benson-Basham cycle, reductive pentose phosphate cycle. A C3 cycle is a series of biochemical redox reactions that take place in the stroma of chloroplast in photosynthetic organisms. It is also known as the light-independent reactions. The cycle was discovered by Melvin Kelvin, James Basham, and Andrew Benson at the University of California, Berkeley by using the radioactive isotope carbon-14. It is one of the light-independent reactions used for carbon fixation. Photosynthesis occurs in two stages in a cell. In the first stage, light-dependent reactions capture the energy of light and use it to make the energy storage and transport molecules ATP and NADPH. The light-independent Kelvin cycle uses the energy from short-lived electronically excited carriers to convert carbon dioxide and water into organic compounds that can be used by the organism. This set of reactions is also called carbon fixation. The key enzyme of the cycle is called Rubisco. In the following biochemical equations, the chemical species exist in equilibria among the various ionized states as governed by the pH. The enzymes in the Kelvin cycle are functionally equivalent to most enzymes used in other metabolic pathways such as gluconeogenesis and the pentose phosphate pathway, but they are to be found in the chloroplast stroma instead of the cell cytosol. Separating the reactions, they are activated in the light, and also byproducts of the light-dependent reaction. These regulatory functions prevent the Kelvin cycle from being respired to carbon dioxide. Energy will be wasted in carrying out these reactions that have no net productivity. The sum of reactions in the Kelvin cycle is the following: 3CO2 plus 6 NADPH plus 5H, 2O plus 9 ATP GLYCERALDEHYDE 3 phosphate plus 2H plus 6 NADP plus 9 ADP plus 8 pi-hexose sugars are not a product of the Kelvin cycle, although many texts list a product of photosynthesis as C6H12O6. This is mainly a convenience to counter the equation of respiration, where 6 carbon sugars are oxidized in mitochondria. The carbohydrate products of the Kelvin cycle are 3 carbon sugar phosphate molecules, or triose phosphates, namely GLYCERALDEHYDE 3 phosphate, Steps. In the first stage of the Kelvin cycle, a CO2 molecule is incorporated into one of 2 3-carbon molecules, where it uses up 2 molecules of ATP and 2 molecules of NADPH, which had been produced in the light-dependent stage. The 3 steps involved are the enzyme Rubisco catalysis, the carboxylation of ribulose 5-bisphosphate RUBP, a 5-carbon compound, by carbon dioxide in a 2-step reaction. The product of the first step is NADol enzyme complex that can capture CO2 or O2. Thus, NADol enzyme complex is the real carboxylase oxygenase. The CO2 that is captured by NADol in second step produces a 6-carbon intermediate initially that immediately splits in half, forming 2 molecules of 3-phosphoglycerate or 3-PGA, a 3-carbon compound. The enzyme phosphoglycerate kinase catalyses the phosphorylation of 3-PGA by ATP. 1,3-bisphosphoglycerate and ADP of the products. However, note that 2 3-PGAs are produced for every CO2 that enters the cycle, so this step utilizes 2 ATP per CO2 fixed. Closing parenthesis. The enzyme glyceraldehyde 3-phosphate dehydrogenase catalyses the reduction of 1,3-BPGA by NADPH. Glyceraldehyde 3-phosphate is produced, and the NADPH itself is oxidized and becomes NADP+. Again, 2 NADPH are utilized per CO2 fixed. The next stage in the Kelvin cycle is to regenerate RUBP. 5 G3P molecules produce 3 RUBP molecules, using up 3 molecules of ATP. Since each CO2 molecule produces 2 G3P molecules, 3 CO2 molecules produce 6 G3P molecules, of which 5 are used to regenerate RUBP, leaving a net gain of 1 G3P molecule per 3 CO2 molecules. The regeneration stage can be broken down in 2 steps. Triose phosphate isomerase converts all of the G3P reversibly into dihydroxyacetone phosphate, also a 3-carbon molecule. Aldolase and fructose 1,6-bisphosphatase convert a G3P and a DHAP into fructose 6-phosphate. A phosphate ion is lost into a solution. Then fixation of another CO2 generates 2 more G3P. F6P has 2 carbons removed by transketolase, giving ERYTHROSE4-phosphate. The 2 carbons on transketolase are added to a G3P, giving the ketose XYLULOSE5-phosphate. E4P and a DHAP formed from one of the G3P from the second CO2 fixation are converted into SEDOHEPTULOSE1,7-bisphosphate by aldolase enzyme. SEDOHEPTULOSE1,7-bisphosphatase cleaves SEDOHEPTULOSE1,7-bisphosphate into SEDOHEPTULOSE7-phosphate, releasing an inorganic phosphate ion into a solution. Fixation of a third CO2 generates 2 more G3P. The ketose S7P has 2 carbons removed by transketolase, giving ribose 5-phosphate, and the 2 carbons remaining on transketolase are transferred to one of the G3P, giving another ZU5P. This leaves 1 G3P as the product of fixation of 3 CO2, with generation of 3 pentoses that can be converted to Ru5P. R5P is converted into RIBULOSE5-phosphate by phosphopentose isomerase. ZU5P is converted into RUP by phosphopentose epimerase. Finally, phosphobulokinase phosphorylates RUP into RUBP RIBULOSE1,5-bisphosphate, completing the Kelvin cycle. This requires the input of 1 ATP. Thus, of 6 G3P produced, 5 are used to make 3 RUBP molecules, with only 1 G3P available for subsequent conversion to hexose. This requires 9 ATP molecules and 6 NADPH molecules per 3 CO2 molecules. The equation of the overall Kelvin cycle is shown diagrammatically below. Rubisco also reacts competitively with O2 instead of CO2 in photorespiration. The rate of photorespiration is higher at high temperatures. Photorespiration turns RUBP into 3-PGA and 2-phosphoglycolate, a 2-carbon molecule that can be converted via glycolate and glyoxylate to glycine. Via the glycine cleavage system and tetrahydrofolate, 2 glycines are converted into serine plus CoO2. Serine can be converted back to 3-phosphoglycerate. Thus, only 3 of 4 carbons from 2 phosphoglycolates can be converted back to 3-PGA. It can be seen that photorespiration has very negative consequences for the plant, because, rather than fixing CO2, this process leads to loss of CO2. C4-carbon fixation evolved to circumvent photorespiration, but can occur only in certain plants native to very warmer tropical climates, corn. For example, the immediate products of 1 turn of the Kelvin cycle are 2-glyceraldehyde-3-phosphate molecules, 3 ADP and 2 NADP+. Each G3P molecule is composed of 3 carbons. In order for the Kelvin cycle to continue, RUBP must be regenerated. So, 5 out of 6 carbons from the 2 G3P molecules are used for this purpose. Therefore, there is only 1 net carbon produced to play with for each turn. To create 1 surplus G3P requires 3 carbons, and therefore 3 turns of the Kelvin cycle. To make 1 glucose molecule would require 6 turns of the Kelvin cycle. Surplus G3P can also be used to form other carbohydrates such as starch, sucrose, and cellulose, depending on what the plant needs. Light-dependent regulation. Despite its widespread names, these reactions do not occur in the dark or at night. There is a light-dependent regulation of the cycle enzymes, as the third step requires reduced NADP, and this process would be a waste of energy, as there is no electron flow in the dark. There are 2 regulation systems at work when the cycle needs to be turned on or off. The orodoxin ferredoxin activation system, which activates some of the cycle enzymes, and the Rubisco enzyme activation, active in the Kelvin cycle, which involves its own activars. The orodoxin ferredoxin system activates the enzymes glyceraldehyde 3-P dehydrogenase, glyceraldehyde 3-P phosphatase, fructose 1,6-bisphosphatase, SEDOHEPTULOSE1,7-bisphosphatase, and RIBULOSE5-phosphatase kinase, which are key points of the process. This happens when light is available, as the ferredoxin protein is reduced in the photosystem I complex of the thylakoid electron chain when electrons are circulating through it. Ferredoxin then binds to and reduces the orodoxin protein, which activates the cycle enzymes by severing a cysteine bond found in all these enzymes. This is a dynamic process, as the same bond is formed again by other proteins that deactivate the enzymes. The implications of this process are that the enzymes remain mostly activated by day and are deactivated in the dark when there is no more reduced ferredoxin available. The enzyme Rubisco has its own activation process, which involves a more complex process. It is necessary that a specific lysine amino acid be carbamylated in order to activate the enzyme. This lysine binds to RUBP and leads to a non-functional state if left uncarbamylated. A specific activase enzyme, called Rubisco activase, helps this carbamylation process by removing removing one proton from the lysine and making the binding of the carbon dioxide molecule possible. Even then, the Rubisco enzyme is not yet functional, as it needs a magnesium ion to be bound to the lysine in order to function. This magnesium ion is released from the thylakoid lumen when the inner pH drops due to the active pumping of protons from the electron flow. Rubisco activase itself is activated by increased concentrations of ATP in the stroma caused by its phosphorylation.